Hello and welcome to my presentation on mutations in protein C-termini associated with disease. This is a proteome-wide analysis of variants that affect a particular portion of the human proteome, which is the collection of every protein in the human body. I'm going to begin by explaining some background information on C-termini and mini-motifs. So what is a C-terminus? To put it simply, a C-terminus is the end of a protein. Proteins are directionally translated from N-terminus to C-terminus, where I've highlighted the N-terminus in red, and I've highlighted the C-terminus in green. The N-terminus is the beginning of the protein, and the C-terminus is the end of the protein. And for the purposes of our project, the C-terminus is the last 10 amino acids of a protein. The C-termini is simply the set of every C-terminus for every human protein. And we are doing an analysis of all of the variants that occur on the human C-termini. Why we're doing this is because the Schiller lab, which is where I am from, has experimentally identified 8 pathogenic variants on the C-termini. And so we're wondering if there are more, we're wondering about their frequency, we're wondering about the mechanism of disease for these variants, and so on and so forth. So now I have to explain what a mini-motif is. To put it simply, a mini-motif is a short peptide sequence that has a particular function inside of a protein. So something like THL, which is a 3 amino acid sequence, can have its own function within the protein. There can also be mini-motifs that only have a function on the C-termini. Actually, 13% of protein coding genes have a functional C-terminus, which means that they have a mini-motif that plays a function on the C-terminus specifically. And these mini-motifs play functions like post-translational modification, protein binding, and protein trafficking. We can divide mini-motifs into 2 different kinds, instance and consensus. An instance is a particular arrangement of amino acids, like the example given here. In this case, this one has a function of post-translational modification. There are also consensus mini-motifs, which have positions of degeneracy, where a position can be occupied by several different amino acids. In the case at the bottom bullet, there's a serine on the left and a serine on the right. Both of those have to be there, but in the middle, there can be any one of the 3 different amino acids listed there. Crucial to this project is the notion of mini-motif loss of function. I have an example protein here where the C-terminus is highlighted in green and a matching C-terminal mini-motif is highlighted between both the green and the yellow. What's highlighted in red is a lysine, which is the M-mod for this mini-motif. The M-mod is the position where the protein is modified. If the M-mod for the mini-motif is altered, then there is a loss of function for the mini-motif and probably for the protein, which is probably going to be pathogenic. I've given the example here where the lysine is changed to an alanine from a K to an A, and therefore the M-mod is lost. Now, there can also be cases where the mini-motif is altered, but the M-mod is not altered, as in the case at the bottom, where a different amino acid is changed to a serine, highlighted with the purple, but the lysine remains intact. These can have a potential loss of function, but they aren't necessarily loss of function. Some of our main hypotheses are as follows: that single nucleotide variants on the C-termini are especially related to disease, that there are many single nucleotide variants that disrupt C-terminal mini-motifs, and that single nucleotide variants that disrupt C-terminal mini-motifs are more likely to be pathogenic. We haven't finished testing all of these hypotheses, but what we have collected is very interesting. The way that we went about this was we took existing big data sets from NCBI, which is the National Center for Biotechnology Information, from the Broad Institute of MIT and Harvard, and from the C-terminome database from the Schiller lab here at UNLV. And we conducted analysis of these data sets using custom Python scripts. In fact, I wrote all of the Python scripts for this. To clarify some terms before I get into the data, a missense variant is one where a single amino acid is altered between the reference and the alternate sequence. In this case, a glutamic acid, which is E, is changed to a serine, which is in S. A nonsense variant is one where a stop codon occurs earlier than normal, so some of the amino acids are not translated. In this case, the last 3 amino acids are basically lopped off. Here's some of the statistics on the ClinVar variants. There are a total of 911,639 variants in the ClinVar dataset, and 31,949 of these, or about 3.5%, are on the C-termini of proteins. 3,963 of these C-terminal variants are missense, and 27,208 of them are nonsense. This is a chart showing the different forms of pathogenicity of these C-terminal missense variants. There are 294 pathogenic, and by far the majority of them are uncertain, which is 2,258. This means that 7.4% of the missense single nucleotide variants on the C-termini are classified as pathogenic. So our hypothesis was correct, that there were many pathogenic C-terminal variants that are missense. Several hundred, in fact. For the nonsense variants, 19,323 of the C-terminal nonsense variants are pathogenic, which is expected. Nonsense variants are much more likely to cause a loss of function in a protein than missense variants are. And so about 70.7% of the nonsense C-terminal single nucleotide variants are classified as pathogenic. This is some frequency data for the missense C-terminal variants. The benign variants are much more common in general. Most of them are located in the 0.1% frequency for the population level. Greater than 0.1% frequency for the population level. Most of the pathogenic variants are located in the 0.001 to 0.01% frequency range or lower. And we would expect this. We'd expect the pathogenic variants to be less common in general because we would expect them to be selected against. Whereas the benign variants, we would not expect those to be selected against. This is a summary of all of the C-terminal mini-motifs in the C-terminum dataset. There are 34,382 C-terminal mini-motifs in total, and almost all of them are instance, 34,310. Only a few dozen are consensus. And most of them also have post-translational modification as their function, 30,562. This is a table that summarizes the mini-motifs where a variant caused a loss of that mini-motif on the C-termini. So potential loss of function of the mini-motif and actual loss of function of the mini-motif. That's what's included here. There are 6,729 of these, and almost all of them, again, are instance, and most of them have post-translational modification as their function. So we've identified 6,729 potential or actual loss of function cases for these mini-motifs being lost. But the real kicker is that we had 112 cases where a missense variant altered the M-mod, the mini-motif was lost, and the variant was pathogenic. Because the M-mod was lost and the variant was pathogenic, that means that the mechanism of disease was that M-mod being lost and the mini-motif losing its function. So we have 112 cases where we know that the mechanism of disease was the mini-motif being lost. This is by far the most important result that we got out of this. So our conclusions, we identified 19,617 pathogenic single nucleotide variants on the C-termini of proteins, with 294 of them being missense. About 7.4% of the missense C-terminal single nucleotide variants were pathogenic, and about 70.7% of the nonsense C-terminal single nucleotide variants were pathogenic. And most importantly, we had 112 cases where loss of function was due to a C-terminal M-mod for a mini-motif being altered. And we're going to continue to analyze all of this data and continue to test our hypotheses. For the funding and acknowledgements, this is simply the NIH grant that has funded this project. And I would like to acknowledge Dr. Joseph Lombardo from the UNLV National Supercomputing Center, who has allowed us to use equipment to conduct these analyses. And I would also like to thank my research advisors, Dr. Martin Schiller and Dr. Jiagong Wu, who have given me the opportunity to engage in research, to perform and work on this project, and eventually to write up the manuscript for this and hopefully get it published. I thank you for listening to my presentation, and I hope you enjoyed.